Hello, so good evening, students. So we'll start the session now. So topic for the day is two-dimensional gel electrophoresis. So today we'll cover 2D gel electrophoresis, its analysis and applications. So let me introduce myself first. My name is Ankit Kumar Malik, a PhD PMRF fellow from the Department of Pharmaceutical Engineering and Technology, IIT BHU. And the course instructor for the same is Professor Sanjeeva Srivastava from IIT Bombay. So let's start from the introduction. So what is 2D gel electrophoresis? So actually 2D gel electrophoresis is a powerful analytical technique used to separate complex mixture of proteins. So it is commonly used in some proteomics to analyze the protein content of cells, tissue or any organisms. So it has two things. So one is it's called why it is 2D gel electrophoresis. Because it has two dimensions. First dimension is isoelectric focusing and second one is second dimension. It's called SDS page. So we'll cover one by one. So first one is isoelectric focusing. So in this dimension, proteins are separated based on their isoelectric points. So also I'll give you the overview of gel electrophoresis. So okay, so let me first explain what 1D and what 2D is, isoelectric and SDS page. So in 1D, which is isoelectric focusing, proteins are separated based on their isoelectric point. There is some all the proteins have some isoelectric point. So in 1D, it got separated according to their isoelectric point, which is the pH at which a protein carries no net charge, which is called isoelectric point of any particular protein. Okay, so this is achieved by applying an electric field to a pH gradient gel. So proteins will migrate in the gel until they reach the region where the pH equals their isoelectric point, at which point they stopped moving, which is called isoelectric point. Okay, so this was the 1D isoelectric focusing, and whereas SDS, which you always know, we have also discussed in the last class as well. So SDS is sodium dodecyl sulfate polyacrylamide gel electrophoresis. So in this step, proteins are denatured and coated with SDS, coated with sodium dodecyl sulfate, which gives them a uniform negative charge proportional to their mass. So when an electric field is applied, the proteins move through the gel. So with smaller proteins migrating faster than the larger ones. Okay. So this is the overview of 2D gel electrophoresis, where the first dimension isoelectric focusing is moving towards decreasing isoelectric point. So here the isoelectric focusing gel is placed on the SDS polyacrylamide gel, whereas in this isoelectric focusing gel is placed on SDS page. So whereas in the second dimension, as you can see, it is in decreasing isoelectric point was there. Okay. So then the basics of 2D gel electrophoresis, which is highly unlikely for proteins to be similar in two distinct properties, which are isoelectric points, protein mass in SDS page, protein complex mass in native page. Okay, so then the advantages of 2D gel electrophoresis. So it has several advantages, especially in the context of proteomics and protein analysis. So first we'll cover one by one. That is a high resolution. So it the two dimensional two dimensional gel electrophoresis can separate thousands of proteins simultaneously based on two independent properties. So one is isoelectric point and another is molecular weight. So this high resolution makes it possible to distinguish between proteins that are very similar in size or charge. Okay, and the second advantage is its detection of protein isoforms and post-translation modification. So in this 2D gel electrophoresis can differentiate between different isoforms of a protein which may vary slightly in charge or molecular weight. And in post-translation modifications, it is effective in detecting proteins that have undergone post-translation modifications. And as these often alter the protein's isoelectric point or their molecular weight. So the preset conditions are the pH ranges, size of the gel, staining methods can be changed to increase the resolution according to the protein and proteomics nature. So this 2D gel electrophoresis is one of the core technology of proteomics. Okay. Then there's one more thing called qualitative analysis. So the intensity of a protein sports on a gel can be quantified to estimate the relative abundance of proteins in different samples. So this can be useful for identifying proteins that are up or down regulated under any specific conditions. And we can also identify protein according to this like by using mass spectrometry compatibility. So in mass spectrometry compatibility, proteins separated by 2D dimensional electrophoresis, which can be easily excised from the gel, digested into peptides, and identified using mass spectrometry. So this makes it a powerful tool for protein identification and its characterization. And some there are some wide application ranges are versatility. So it is versatile, which is applicable to wide range of samples, including whole cell lysates, some tissue extracts, and you can say body fluids. So this is useful in various research fields, including cell biology, medicine, and biotechnology. Okay. So there are some while 2D gel electrophoresis is a powerful technique, it has several limitations as well that can affect its applicability and effectiveness in certain situations. As you can see, it is hard to resolve in its poor resolution, sample size. See, the resolution is the advantage and limitation both. So we'll cover one thing one by one. So it has sample size which protein cannot be amplified like DNA, and some automation process difficulties are also there. So we'll cover one by one. So also there is one thing, limited protein solubility. So it is hydrophobic, whereas in the hydrophobic proteins, membrane proteins and other highly hydrophobic proteins are difficult to solubilize and separate using 2D gel electrophoresis. So these proteins often precipitate or do not enter the gel effectively, leading to incomplete analysis of the proteome. Okay, and it has some dynamic range limitations as well, whereas it struggles to detect proteins present at a very low concentration, especially in the presence of highly abundant proteins. So the dynamic range of protein detection is often limited, which can result in underrepresentation of any low abundance proteins. Okay, and it has some reproducibility issues as well, and some limited analysis of very large or very small proteins, like separating very large proteins above 200 kilodaltons or very small proteins below 10 kilodaltons. So these proteins may not resolve well or may be lost during the 2D gel electrophoresis process. And it has some difficulties with highly basic or highly acidic proteins. So, like proteins with extremely high or low isoelectric points can be challenging to separate using this technique. So, as they may not focus well in the isoelectric focusing step. Okay, and there's some more limitations like protein degradation and modifications during the process. So, in gel degradation, so proteins may degrade or undergo unwanted modifications during the lengthy process of 2D gel electrophoresis, so leading to its artifacts or loss of protein spores. Okay, and there are some like limited identification capability, some data analysis challenges are there, and then some alternative techniques are there like emergence of mass spectrometry based proteomics with advances in MS and LCMS as well. So throughput, the dynamic range and the use of 2D gel electrophoresis has declined in some of the areas of proteomics. Okay, then there are some certain steps we'll follow for the this technique. We'll just take break for 2 minutes. Wait. So 2D gel electrophoresis steps. So it has a series of steps to separate proteins based on their isoelectric point, that is pi. So and molecular weight as well. So here, as an overview of the main steps involved are sample preparation, 1D separation. As I told you earlier, it is 1D is isoelectric focusing, and 2D is SDSP. By combining both the things, it will come the 2D gel electrophoresis by combining both the separation steps like isoelectric plus SDSP. Then detection of spots, analysis of protein spots. Okay, so we'll cover one by one now. So the first, as you can see, sample preparation. So here, proteins are extracted from the samples. The samples must be any cells, tissue, or any body fluids. So using a lysis buffer that typically contains detergents, reducing agents, and protease inhibitors. So this ensures the proteins are solubilized and protected from degradation of any kind of degradation. So then protein quantification as well. So the total protein concentration is measured to ensure consistent loading across the sample. It will come in sample preparation as only. So second one is first dimension separation. So here there is one term called IPG, uh, stands for immobilized pH gradient. So in further class I'll use that IPG term. So the rehydration of IPG strips. So strips are rehydrated in a solution containing the protein sample. So the IPG strip has a pH gradient immobilized in a gel matrix. So the rehydrated IPG strips with the protein samples are placed in an isoelectric focusing device. So thereafter an electric field is applied, causing proteins to migrate along the pH gradient until they reach the point where their net charge is zero. So at this point, they stop moving and focus into sharp bands. Okay, so and after this 1D dimension, the second is equilibration of the IPG strips. So the IPG strips are equilibrated in a buffer containing a reducing agent like DTT to break disulfide bonds and alkylating agents as well. So the alkylating agent, as you can see, for example, is iodoacetamide is there, which we can use as an alkylating agent. So which prevents reformation of the bonds. So this step prepares the proteins for the second dimension, ensuring they remain fully denatured. So after this, we'll go to the second dimension separation, which is SDS page. So here, polyacrylamide gel is prepared for the second dimension. So the gel may have a uniform or a gradient acrylamide concentration depending on the desired resolution. Okay. So first we'll do some SDS coating. The IPG strip is placed on the top of SDS page gel. So SDS, as I've already told you, it is a detergent that coats protein, giving them a uniform negative charge proportional to their molecular weight. Okay. So if you don't know the word electrophoresis means, so electric field is applied perpendicular to the first dimension, causing proteins to migrate through the gel. So since all the proteins have a similar charge to mass ratio, so due to sodium rhodocyl sulfate, so they are separated based on their molecular weight, with smaller proteins moving faster through the gel. Okay, and so the next in this SDS page, it will come protein visualization is there. So in protein visualization, we'll look for staining, cosmic brilliant blue, then silver staining, fluorescent staining, and de-staining process are there. So then followed by some image analysis as well. So in which gel imaging will be done. The stained gel is scanned or the photograph to create any digital image. So after that, we'll go for spot detection and quantification. So there's some software used to detect and quantify the protein spots. So the intensity of each spot correlates with the amount of protein present in the sample. So then after that, one thing is called comparative analysis. So like if we compare different sample like diseased or healthy, like control, negative control or some positive control. So the gels are compared to identify differences in the protein expression. Okay. So then this I've already covered. So the just there's some contaminants and the removal procedure are there. So like there are some particles, some charged molecules like salt buffer components are there. So there put an poor focusing effect on the sample, which can be removed using a dialysis or a gel filtration. Then SDS has the protein move towards positive electrode, and it can be removed by acetone precipitation, as well as for the DNA and particulate matter as well. Then protein solubilized are there, some lysis solution compositions are there as you can see in the chart. So we have some urea detergents, reducing agents, as I told you, DGT. Then ampholites are there with their final concentration and their function. Okay. Then there's a thing called sample preparation protocol. So we'll explore this thing, the entire flow chart. So sample preparation is a crucial step in 2D gel electrophoresis as it directly affects the quality and resolution of the protein separation. So here is a general protocol for preparing protein samples for 2D gel electrophoresis. So the first thing, we required some materials before starting this protocol. So as I've told you in some earlier slides, so I'll give you some example of some materials needed for this protocol or any sample separation. Like we need some lysis buffer, protease inhibitor cocktail, then DNAs and RNAs, NDTT, which is dithiotherietol, then some solvents like acetone or methanol or chloroform from for protein precipitation, or which is optional. Then centrifugation technique will be there. Then sonicator or any homogenizer. Then then IPG strips. Yes, IPG strips are also there. So the first is cell or tissue sample collection. So we'll collect the biological samples from any cells, tissue, or any body fluids. So for cells or tissue, snap freeze the samples in liquid nitrogen and store them into minus 80 degrees Celsius until used to prevent the protein degradation. Okay, then the second step is perform lysis. So here lysis buffer preparation will be there. Like we use some 7 molar urea, and which that agent that helps solubilize proteins. Then we use some 2 molar thiourea, which enhances the solubility of hydrophobic proteins. Okay, then some more called 4% chaps we say in our layman language, which is CHAPS. Then 1% DTT, which is a reducing agent to break disulfide bonds in the proteins. And some protease inhibitor cocktails also we can use, which are used to inhibit the proteolytic degradation during lysis process. Then the next thing is lysate. So here we can add an appropriate amount of lysis buffer of the sample, which is usually 10:1 buffer to tissue weight, or you can use 1 ml per cells. Okay, so followed by this, homogenize the tissue or cell sample using a homogenizer, or you can use sonicator or vortexing to ensure complete cell disruption. After that, we can incubate, and when we can incubate that lysate on ice for 30 minutes to allow through the protein extraction. Then it can be followed by the next step, which is a removal of insoluble material. So we can centrifuge the lysate at high speed for 15 to 30 minutes, like at 4 degrees Celsius. Then pellet cell debris, then DNA and other insoluble material, which can be carefully collect the supernatant, which contains the solubilized proteins without disturbing the pellet. Then followed by that step, some protein quantification is there, from which we can measure the protein concentration using the BSA or Bradford assay, which ensures that the protein concentration is within the range, within the suitable range of 2D, that is 1 to 2 microgram per ml. Okay, then the last is sample cleaning, which is an optional thing, sample cleanup. So to further remove the interfering substances such as salts, nucleic acids, or lipids present in the protein samples by using a commercial 2D cleanup kit if necessary, if it is required. Okay. Then after that, we can store the samples. So if not immediately proceeding for further processing, so the store we can store the prepared protein samples at minus 80 degrees Celsius. Then to avoid repeated freeze thaw cycles, which can degrade proteins. Then the last is rehydration of IPG strips. So by combining the protein sample with a high rehydration buffer containing the same components as the lysis buffer, so we can load the sample onto the IPG strip, ensuring uniform distribution. The followed by the amount of protein loaded typically ranges from 100 microgram to 1 mg. So depending on the sensitivity of the staining method to be used later. So lastly, we can seal the IPG strip in the rehydration tray and incubate at room temperature for passive rehydration. Usually we can keep it for overnight. Okay. So there are some points which I've already told you, like separation of basis, isoelectric point, not molecular weight, then which requires high voltages near about 5000 watts, and also it requires a long period of time, that is 10 hours. So presence of pH gradient is critical. Uses are lights to establish pH gradient. So this is a diagram you can see. So here an isoelectric point is the pH of a solution at which the net charge of protein is zero. Then in electrophoresis technique, there is no motion of the particles in an electric field at this electric point. Okay. So this I have already told you. Like it has some soluble amphalites, which ranging pH ranges from 4 to 8. Any slides are not changing. Now I'm at 14th slide. So now I think it will change. Or wait, I'll reshare the screen. So now is it visible? Like now is it changing? Wait, I'll go to the 14th slide, from which where we left. So now we're at 14th slide. So there are some pH gradients like soluble ampholytes. As I've already told you, it varies from batch to batch, and it has a pH range about 4 to 8 at equilibrium. And it has some immobilized pH gradient which is called IPG strips. It has some acrylamide derivatives with charged change and stable and irreducible gradients and higher mechanical strength, higher protein loading capacity. So these are the IPG strips which I have told you. It has some broad range. It has some three types: broad range, medium range, and narrow range. Depending upon the experiment, we can use any of this strips range. So these are some pictorial things for isoelectric focusing. So which load samples which per group, then peel off the protective film from the strip. Then we can in the second image, as you can see, you can place the IPG strip gel facing down in a groove, then rehydrate overnight more than 22 hours at a room temperature. So these are some protocols or parameters are there, so which can slowly increase the voltage and apply a high voltage at and to obtain sharp narrow zones of the proteins, so which has some broad range and narrow range. So there are some different protocols according to their watts and requirement. Okay. So this all I've told you. So second thing is called two second dimension SGS page steps. So here we'll start the SGS page things. So SGS page stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. Okay, which is widely used technique for separating proteins based on their molecular weight. So it is a key method in protein analysis and is often used as a second dimension in 2D gel electrophoresis, followed by isoelectric focusing. So it has some key components like SDS has some different things, and electrophoresis came from joining two words by combining two words SDS and page. So what SDS is? So SDS is a detergent that binds to protein, so denaturing them and giving them a uniform negative charge. So this charge is proportional to the protein's length. So meaning that when an electric field is applied, the proteins will migrate through the gel primarily based on size rather than the charge. And on the other hand, page, that is polyacrylamide gel. So it is a mesh-like matrix that acts as a molecular seal through which proteins migrate during electrophoresis. So the size of the pores in a gel primarily based on size rather than the charge. Okay, so how it does work, how SGS page works. So first we'll prepare the sample. So there are some steps. So first we'll prepare the sample. Just wait for 2 minutes. So we'll prepare the sample. So in that proteins are mixed with the SGS and a reducing agent like GTT or beta mercaptoethanol is there. So the SGS binds to the proteins, denatures them by disrupting non-covalent bonds and imparts a uniform negative charge. So the reducing agents break any disulfide bonds, ensuring the proteins are fully denatured. Then followed this, we can load the gel, which you can see the loading the gel. So here we can prepare protein samples are loaded into the wells of the page. That is polyacrylamide gel. So a molecular weight marker or any ladder which contains proteins of known sizes is also loaded for any references. Okay, then we'll go for some electrophoresis techniques. So, and after this, an electric current is applied across the gel, causing the negatively charged protein to migrate towards the positively charged electrode. Smaller proteins move faster and further through the gel matrix, while larger proteins move more slowly. Okay, so then we'll go for the separation. So, as a result, proteins are separated by size, with smaller proteins appearing lower in the gel and larger proteins higher up. Okay, then followed by the staining. So after electrophoresis, the gel is stained using a dye. Like we can use any dye, like Cosmase, Brilliant Blue, or Silver stain are the most common dyes which can be used after electrophoresis. So these dyes can be used to visualize the separated proteins. Each band represents a protein or a group of proteins of a similar molecular weight. So after all these steps, we'll go for the analysis. So the stained gel is analyzed to determine the molecular weight detection. Wait, there are some photographs which you can see that cosmic stains and silver stains are there, which we have used after the step electrophoresis. So we'll then go for analysis. So the stained gel is analyzed to determine the molecular weight of the proteins and to compare the relative abundance of proteins between different samples. Okay. So there are some more stains are like copper stain and cyprose fluorescent is there. So there's some troubleshooting as well, horizontal streaks. So in horizontal streaks and vertical streaks, so samples are not completely solubilized prior to the application on the IPG strip, and sample poorly soluble in dehydration solution, ionic impurities and some ionic detergents as well. So whereas in vertical streaks, there are some insufficient equilibration, insufficient SGS is there. Okay. So there are some application as well of SGS page like protein identification, purity assessment, protein quantification, and Western blotting as well, which we can use in by STS. So after this, some softwares, 2D gel softwares are there by which we can analyze these protein spots. So there are some screenshot or interface of that software which I have taken screenshot for you. So you can do Swiss 2D page, and there are some commercial softwares are present on online which you can use for the analysis of sports by name Melanie 4 Image Master 2D Elite and 4 ATEX 2D Advanced and PD Quest 6.1. So these are the links given below to download the particular commercial software. So then common software features. So how the softwares can be useful or beneficial for the analysis of sports. So it gives some image contrast and coloring. We have some gel annotation features are there with sport selections and marking can be used by using that particular annotation. Then some automated peak clicking are there. Some sport area determination is there, which we call integration, and matching 2D gels to comparing further comparing two or more gel metrics, and stacking, aligning, and comparing of the gels are also present in the software, which is very useful and helpful for the analysis of the things. Okay, then there are some analysis things are there machines which is used for the excision the sports of interest, by which we can pick up the protein gel sports from gel, like it can be automatic or any manual things. So in this machine there is excision in the sports of interest. So it contains the in-gel digestions by using the washing process, dehydration and drying process, trypsin digestion, extraction, desalt and concentrate peptide. Okay. So there are some analysis identification. Eluted protein spots are there called multi TOF, which it is a layman thing, which stands for matrix assisted laser desorption and ionization time of flight, and mass spectrometry and peptide mass fingerprint. Then now we'll go through some applications of 2D gel electrophoresis. So it has a wide range of application in various fields, biological and medical research. So here are some of the primary applications of 2D gel electrophoresis. So first we can use in proteomics. First major use of 2D gel electrophoresis in proteomics. So here we can use some protein profiling, in which 2D gel electrophoresis to use in proteomics to profile the entire set of proteins, a single unit of proteome, so which is expressed by a cell, organism, or any tissue. It helps in identifying differences in protein expression under various conditions such as disease states or environmental stress. So it also gives some comparative proteomics. So by comparing 2D gels from different samples, like I told you, like negative control, positive control, untreated, treated, healthy or diseased. So the researchers can identify proteins that are differentially expressed, which may serve as a biomarker or therapeutic targets. And there is a thing post-translational modification analysis. So here, 2D gel electrophoresis is particularly effective at separating protein isoforms that differs due to post-translational modifications, such as phosphorylation, glycolization, acetylation. These modifications can cause changes in a protein's isoelectric point or their molecular weight, so resulting in distinct spot on the gels. Okay, so there's one thing called mapping modification sites, so which can be coupled with mass spectrometry, so which allows the 2D gel electrophoresis for the identification and characterization of specific PTM. Like PTM stands for post translation modification on proteins. Okay, Then there is followed by the biomarker discovery. So here 2D electrophoresis is used to identify potential biomarkers for diseases by comparing protein expression patterns between healthy or diseased samples. So proteins that show significant changes in abundance or modification may serve as a diagnostic or prognostic markers. So it can also be used for protein purification. So there are some two terms are there in protein modification, protein purification. So the first one is isolation of proteins, and the second is enrichment of low abundance proteins. So which can be used. So 2D gel electrophoresis also used as a purification step to isolate specific proteins or protein isoforms from complex mixture, which can then be further analyzed or used in functional studies. So also it can help to enrich low abundance proteins that might be masked by more abundant proteins in one dimensional separations. Okay, and it also has some functional proteomics derivatives as well. So here, which this study can changes in some proteome in response to some, you can say, different functional states, such as during cell differentiation, stress responses, or any metabolic changes. It can also use in some microbial proteome, microbial proteomics, you can say. So we have 2D gel electrophoresis is used to analyze the proteomes of bacteria, of any microbes like bacteria, fungi, or any other microorganism. So which helps in understanding the microbial physiology or pathogenicity and responses to any of the environmental changes. Okay, so it can used in some virulence factor as well. So in pathogenic microorganisms, so here is a flow diagram, a flow chart for the clinical specimen by differentiating the normal cells and tumor cells by electrofocusing in SGS page by using 2D electrophoresis technique. So also there are some tumor cells and normal cells. So tumor biomarker identification is there. So which is extensively used in cancer research to identify proteins that are differentially expressed in tumors compared to normal tissues. Okay, so these proteins may serve as cancer biomarkers or therapeutic agents or therapeutic targets. So by using the understanding of tumor heterogeneity, so it helps in studying the heterogeneity which within the tumors by analyzing different regions of a tumor or comparing primary tumors with metastasis. Okay, then it can be also used for some neurosciences and agricultural research as well. So, so in neurodegenerative disease research, so with such as Alzheimer's, Parkinson's, or Huntington diseases, it can help identifying proteins that are involved in the disease process or that serve as a potential biomarker for early diagnosis. Okay, there are some different applications environmental and agricultural research, evolutionary biology, pharmacology, and toxicology as well. Okay, so at last I have given the a proper schedule if you are planning to do any to this 2D electrophoresis experiment. So we have to schedule and the entire process like a week before or a day before. So here's some schedule, my schedule. So on the day one, we'll prepare the sample preparation and IEF, then which contains the load protein samples onto the IPG strip, then run the experiment by about for 24 hours, then polyacrylamide gel casting. So this was all for the day one. Then followed by day two, which in day two will equilibrize the IPG strip and running SGS page. So this day will remove the IPG strip from the machine. So we'll equilibrium the IPG strip. So then after followed by the equilibrium, the IPG strip onto SGS page, and we can run the SGS page overnight. Followed by this, so at day 3, we'll scan the images, we'll analyze the images. So here we'll remove the gel from the cassette. So the stains are the gel by ruby or silver casting, any staining. Okay, so we'll scan the gel image, we'll analyze the image, and after this, so day 4, in gel digestion, multi-time of flight and database search will be done. So in this, so in that day, in 4th day, we'll pick the protein gel spot from the gel. So we'll do the in gel digestion, followed by spot the sample onto multi-chip and multi-top analysis and some database search will be done. So I've given here some 2D database of 2D maps. So there are some names of the maps and their URL and their images of sample gels are given here. As you can visit this, when you visit this URL, you can find some Swiss 2D page, then her 2D page, HSC 2D page, Metro page, VS 2D page, and some Max Planck Institute are there. So these are the links for this particular database of 2D maps, which are done earlier in the history of research. So there are some protocols, online protocols, which you can read for the 2D electrophoresis. So this is all for the day. Thank you guys. So you guys can leave. We'll meet next week.